Hi everybody, I am Dr. Bijaya Mishra, retired Associate Professor Botany from Govt of Odisha, Higher Education. I welcome you all to the channel Art of Learning Botany. Now you have learnt about the development of endosperm, the post-fertilization changes in angiospermic plants. Today we will discuss about the development of uh, embryo in angiospermic plants. You know, the embryo is a basic thing from which the main plant body comes out. So this is called tiny plant compressed inside the ovule or inside the seed. When they mature, the ovule mature to seed. So this embryo, due to presence of embryo, these plants are called embryophytes. So starting from the bryophytes, there are embryo present. But all the embryo bearing plants or the embryophytes don't produce seed. Only the angiosperms and the gymnosperms they produce seed. So they are called the spermatophytes. All embryophytes are not spermatophytes, but all spermatophytes are embryophytes. That is in one system of classification. Bryophyte is an embryophyte, but this is not a spermatophyte. Pteridophyte is an embryophyte is not spermatophyte, but angiosperms and gymnosperms are embryophytes and spermatophytes by their seed. Okay, anyway, we'll be discussing the embryo development in angiospermic plant, structure and development of embryo in the angiospermic plant. So, now what is an embryo? So, embryo is a post-fertilized product that develops from the zygote inside the ovule. It is a compressed tiny plant packaged inside the ovule or inside the seed. Some say it to be the germ of the seed. Some say it to be the germ of the seed is a tiny plant. It is some say to be a germ of the seed. Right. Now, so let us see the structure of the embryo. So embryo structure is basically same in all angiospermic plants. So it is based on a common body plan. And a standard embryo, a typical embryo consists of an embryonic axis, then cotyledons. The number varies. It is 2 in dicotyledon and 1 in monocotyledon. Then the upper part of the embryonic axis above the cotyledon is called the epicotyle region. It terminates to the plumule, which gives rise to the shoot system or the embryonic shoot. You can say plumule is called the embryonic shoot. And the lower part below the region below the cotyledon is called hypocotyle, and that gives rise to the embryonic root system. In monocots, the covering is there over the plumule. It is called coleoptile. In dicots, in monocots, there is a covering of the radicle. On the hypocotyle covering of the radicle, it is called the coleorhiza. Now, the plumule gives rise to the shoot system or branches, and the radicle gives rise to the root system. The covering, that is coleoptile and plumule, and covering on the radicle, the coleorhiza, is not found in dicots, right? So this is the standard structure of the embryo in angiosperms. Okay, the two cotyledons present laterally and the plumule in dicot, but one cotyledon present on one side of the plumule in monocot. Now let us come and see the development, the structure. Yes, the structure of the dicot embryo. You see how the embryo is packaged inside the seed, and you see it's very curved. These are the two cotyledons. These are the two cotyledons, and this is the radical region of the lower part of the hypocotyle region, and this is monocot. This is the endosperm, and this is the red portion. The pink portion is the embryo. Okay, this is the cotyledon, and this is the plumule region, and the radical region with the coleorhiza and coleoptile. Now come to the development of the pattern of development in angiosperms embryo. Now the development of embryo is called embryogenesis or embryogeny, and it develops from the zygote. So embryo develops only if the endosperm has developed to certain extent. If endosperm, there is no development of endosperm, there is no development of embryo. Embryo will not develop. So endosperm development ensures provision of food to the coming child or the embryo, right? Now the development of embryo from the zygote is called zygotic embryogenesis, and there are some changes before the zygote develops to embryo. So what are the changes? There is a polar orientation. Now the nucleus of the zygote will move towards the apical region or the top region, the chalazal end. Now the chalazal end of the ovule becomes the anterior end, and the micropylar end will become the posterior, the basal end. And cytoplasmic contents come to the basal end, the nucleus move to the chalazal, the upper end, which is now the apical pole, and is called the basal pole or the basal end. So the polar orientation starts. The polar orientation of the plant starts itself from the zygote division or the formation of embryo. Right. Now let us see the development of embryo, how it was the pattern of development. Now the pattern of development of embryo in angiosperm is almost similar. Basic plan is similar. The body plan is similar. Now the embryo develops two phases. One is the morphogenesis phase, where by division it will differentiate into different parts of the embryo. Then maturation phase. They will mature to the different organs. Now the zygote, which is now polarly oriented, divides by a transverse division, and this division you can see here divides by transverse division to form a basal cell, which is toward the lower pole or the micropole region. Posterior pole, you can say lower pole. Basal is the larger cell, and the small one is called terminal cell, the apical cell, which gives rise to the embryo proper. This remains from the chalazal or the apical region. Actually, the proper embryo is formed from the terminal cell, and the basal cell gives rise to the suspension. Right? It gives rise to the suspension that pushes into the endosperm and brings nutrition to the embryo. Now, these two cells develop simultaneously. Both apical cell or the terminal cell and the basal, they develop simultaneously. This will develop and that will develop. So they are dividing together. So not that the terminal cell develop fast or the basal cell develop fast. They divide together, simultaneous development, synchronized development, right? Now, coming to the next slide, we will see the development of the basal cell first. We will see the development of the basal cell. Now, the basal cell, which is present towards the lower pole or the basal pole, now divides by transverse division, repeated transverse division, to form 6 to 8 cells, long filamentous structure, and the distal cell from the apical cell, the last end of the cell of this basal cell division is called the hosterium. So this is called a suspensor. Basal cell gives rise to suspensor, and each cell derivative of this basal cell is called suspensor cell, and the distal cell, which is present toward the end, is called the vesicular cell and is termed as hosterium. And this cell, which is very close to the apical region, but the derivative of the apical cell is called the hypophysis. Okay, the hosterium pushes into the endosperm to bring nutrition to the growing embryo. So this is the development. You can see here. I will zoom it, and you can see. You can see it here. Yes, you can see. This is the hosterium. This is total suspensor. This is total suspensor. Each cell, this one is called suspensor cell, and this is the derivative of the apical cell, and this last cell, this one is called the hypophysis. Okay. Now coming to the development of the apical cell. Apical cell now develops to the embryo proper. The apical cell now develops to the embryo proper. Now how it develops? Now the apical cell, now you see this suspensor, now the apical cell, now it divides either by longitudinal division or by transverse division. So there are different types of embryo development in angiosperms. Now depending the type of division of the apical cell and the derivatives from where they are coming. Now you can see here there are different types. There are 5 types of embryo development. One is crucifer type, another is asteroid type, the other one is solanite type, another is chenopodiad type, another caryophyllid type. But The crucifer type is considered as the typical embryo development in angiosperms, and it is the typical plant. The typical embryo development is studied in Capsula bursa pastoris. So here what is happening now? The embryo, the apical cell. Let us say this is the apical cell. Now the apical cell first will divide by a longitudinal division. Two cells are formed. One of this side longitudinal one. Okay. Then it will divide by another longitudinal division right angle the first one. So if this one, the first one is this, then it will be like this. So 4 cells are formed. After that you will divide by transverse division. Transverse division. So you will find upper 4. So 8 cells are formed. This is called octet stage. The upper 4 tier and the lower 4 tier. So upper tier of 4 cells are called the epibasal cells, and the lower 4 tiers are called the hypobasal cells. The embryo, the upper epibasal cells will give rise to the plumule and the shoot system and the epicotyle region, where the lower part of the cell is called the hypocotyle region and the root system and root parts. So further division of both epibasal cell and the hypobasal cell, it gives rise to different parts of the embryo. So this is at the pro-embryo stage up to the octet stage. Further division will give rise to the other stages, like you will find heart-shaped embryo. Then you will find, let us see this one. You see, let us see the diagram here. You see, this is on the octet stage. You see, this is the suspension, and the octet stage will be formed here, and further we will give it a globular stage, a ball-like structure has been formed. Now in the next stage you will find different stages. Let us see the different stages. Yes. Now you see, here it is a globular stage. Now due to the development of the cotyledons develop very fast. The plumule remains little depressed, so it becomes the heart-shaped structure. Then due to elongation of the suspension and the growth of the cotyledon, to adjust them it becomes curved. Okay, so to adjust inside the sheet. Now coming to the next slide, you will find the how the structure looks like. Up to this is called the proembryo, up to the octet cell globular stage. Now you see this is a heart cell structure, this is a heart stage, torpedo stage. If you see the vesicle here, the ostomium. Now this is a, due to growth of the cotyledon, it becomes a walking stick stage. Walking, the old man's walking stick, carved. So walking stick stage, and further growth of the cotyledons and the embryo gives a horseshoe stage, horseshoe shaped stage. That is packaging, a good packaging occurs inside the sheet or the ovule. Okay. So this is how the embryo develops. We are not going to details. Up to the octet stage you can write, and the upper epibasal cell gives rise to the whole of the epicotyle and the cotyledons develops. Now the radical, the hypophysis region, that the hypobasal cells give rise to the root regions. Okay. Now coming to the other, now we'll find the monocot embryo. Yes, you see here how it is packaged properly. How the embryo is packaged inside the sheet or inside the ovule. Ovule when matures becomes the sheet, right? So here you find the suspensor is very close to the ostomy, very close to the micropyle, the basal pole. Okay, this is the anterior pole. The cotyledon remains the anterior pole. It is inside the endosperm, and endosperm is this region is present. This is the covering of the ovule. So how it is oriented to develop, to germinate to a little plant. Okay. Now coming to the monocot embryo. Monocot embryo is very similar to that of the dicot embryo. It is a corn sheet. Now here you find the major part is the endosperm, and this pink big part is called the scutellum. It is actually the cotyledon. It's a papery cotyledon in monocot is called scutellum. It increases the surface area so that absorption of food can come from the endosperm. Then this is the plumule region, which is covered with the coleoptile, and this is the root apical region or the radical region covered with the coleorhiza. So okay, so this will give rise to only, it contains only one cotyledon. So it is called monocot and laterally present to the premium. Now coming to the difference between monocot embryo and dicot embryo. Now next slide we will see the monocot embryo and dicot embryo development. Difference. In monocot embryo there is only one cotyledon. In dicot there are two cotyledons. The plumule occurs distally, and here in monocot the plumule occurs laterally, and the coleoptile is absent in dicot. Coleoptile is present. Coleoptile is covering of the plumule. It is present in the monocot. Coleorhiza is the covering the radicle, absent in dicot. It is present in the monocot. Scutellum, that is the zinc cotyledon, papery cotyledon, is absent in dicot and present in monocot. The suspense is large in dicot and more smaller in monocot. Okay, so we have discussed up to now the structure of the embryo, what is a pro-embryo, what is the basic pattern of embryo development, and what are the stages, different stages of the embryo, the globular stage, the hard stage, then the torpedo stage, then the walking stick stage, then the horseshoe stage, and what is the polar orientation. Plumule will give rise to the shoot system, the radicle will give the root system. So I have all discussed in this portion. Okay, now we'll see a self check for yourself the questions. Now the questions is fill up the blank questions here, and you can see fill up the blanks with most appropriate words or answer. The portion of the embryonic axis above the cotyledon is called embryonic axis above the cotyledon is called epicotyle. Portion of the embryonic axis below the cotyledon is called hypocotyle. The hypocotyle terminates to radicle. The epicotyle terminates to plumule. The plumule is therefore a part of what? Epicotyle. Definitely. Now coming to the sixth question. The suspensor is a derivative from the division of the dash cell of the two-celled embryo. Basal cell. The two-celled embryo means apical and basal cell. Suspensor is the lower point. So it is called the derivative basal cell. The covering the radicle is called, the covering is found in monocot. So its covering is called coleorhiza. The lowermost cell of the suspensor close to the apical cell derivative is called hypophysis. The distal is called the hosterium. The close to the apical cell is called the hypophysis. The uppermost vascular cell of the suspensor or distal cell of the suspensor is called hosterium. The hosterium is found towards the dash region of the ovule, the micropilar region of the ovule. Now coming to the next question, we will get the short answers. Now let us see the short answer questions. Yes. What is a hypocotyle? Hypocotyle is the lower part of the lower region of the proembryo below that of the cotyledon. It gives rise to the radicle and the root system. What is an epicotyle? Epicotyle is the embryonic part above the cotyledon in a proembryo. It contains plumule, terminates to plumule and gives rise to the shoot system. What is a hosterium? Hosterium is the distal cell or the terminal last cell of the suspensor, is also called a vesicular cell. It brings nutrition from the endosperm. What is a scutellum? Scutellum is a papery cotyledon found in monocot. It increases surface area for absorption of nutrition from the endosperm. What is a radicle? Radicle is the part of the hypocotyle, a terminal part of the hypocotyle that develops to the root and root other parts. It is covered with coleorhiza in monocots. Now what is a coleorhiza? Coleorhiza is a covering of the radicle in monocots, not found in dicots. What is a coleoptile? Coleoptile is a covering of the plumule in monocots. It is not seen in dicots. Which portion of the proembryo develops to shoot system and let me see, yes. Which portion of the develops to shoot and root? The epicotyle region of the proembryo develops to shoot system. It contains plumule and cotyledon that develops to the shoot system. Whereas the hypocotyle region of the proembryo gives to the root system, which terminates to the radicle. Now, what write three differences between the dicot embryo and monocot embryo. So there are many different, 5 or 6 differences are given. So you can write any three. For that, first difference must write that number of embryo cotyledon. In dicot, 2 cotyledons, monocot 1 cotyledon. Position. The two cotyledons present on the two lateral sides, either side of the plumule, and is present on lateral side of the plumule. Right. Then scutellum is absent in dicot, scutellum is present in monocot. Coleoptile, coleoriza is absent in dicot, is present in, they are present in monocot. Suspensory larger in dicot, is smaller in monocot. So you can write all these points. So why the radical is present towards the hypocotyl present towards the lower side, towards the micropyle, because it will give rise to root. So root should first come, shoot will not come first. First root will come when the seed germinates. So it's present towards the micropyle region. Okay, so we have seen the embryo development in details here. How what is an embryo, how it develops, which part will give it to shoot system, which part will give rise to the root system, and what is the difference between monocot root and dicot root. Okay, so I hope you have studied well, I have understood the lesson well, and like it, subscribe the channel, and you can subscribe the channel and see our further videos.